Harry, thank you very much for accepting to this. We need to say that you are a merit research project. So could you please tell us how you've gone through your process?
So first of all, my research question is how can scientists grow organs in laboratories and in animals, and is this technology ethical or not? So first of all, I did the proposal in which I attempted to refine the topic. At first, I only know I wanted to do a topic on growing organs, but not exactly this topic. So I went through the refinement process discussing with my research project teacher, and she helped me to come up with this topic. And then capability of my research, I picked literacy and information communication technology as my capabilities. Literacy because I will have to do a lot of online research and it will be important. And information and communication technology because I will have to conduct interviews through online sources. Then I wrote on ethical of research and planning of my research process, which in the end assisted me in coming up with a timeline schedule for my research. Over here's a mind map. It's quite big, but so I did an annotation on the challenges of growing human organs because that's the main topic I'm going to do for my outcome. And then Lotus diagram did a similar topic as the mind map, but it's just more vague. And I feel
so what's the difference between your mind map and your Lotus diagram?
Lotus diagram talked about the main topics, whereas the mind map talked about the topic I'm going to do.
And why it's color-coded?
Color-coded so I can understand which part it elaborates on which topic. For example, there are the challenges of stem cell research and capabilities. This way I don't get them mixed up. I filled the bottom right corner with reflection on the mind map. And
when did you do that pink part?
After I finished my outcome. This is towards the end of my research, so I could go back and just see what helped and what worked.
Lovely.
And then reflecting on my chosen capabilities. This was done towards the end of my folio, so I could go back and evaluate what capability I developed the most and what new capabilities I acquired. I highlighted these sections which talks about the main points on what literacy skill was improved and what information and communication technology was improved. And then there's an outcome timeline, which I wrote my outcome according to. It came in handy, but the word count in the end had to be changed. Moving on to research, this is my secondary online research where I chose some articles and annotated them.
So let me take a good picture of this.
Yep.
So it's very busy. And can you explain to us what these colors are there?
The colors are, the green one are really good information to include in my outcome. The yellow ones are information I found harder to understand and require other secondary research to prove. Blue ones are the quotes I decided to use in my outcome. And the red annotations, although they are few, they're information I really don't understand. I need to research more on.
So you did a huge research, but we see only 2 pages of secondary sources. Why?
Because these 2 pages helped me the most.
Okay.
Most of my information came from this huge article that the American Health Association did. And it really helped me in my outcome. And I thought to annotate this one would be most relevant to my outcome.
Lovely. And what are these here? These
This
are
one
videos.
is also secondary research, but they're videos on YouTube.
Why you use them? Why you thought them were credible and reliable?
For example, some of the sources are from TED Talks, which are known to be quite credible, and other ones are actually news reports. Because my topic is a fairly new scientific topic, a lot of articles I wrote, but also a lot of video format information. So I thought annotating them would help greatly to write my outcome.
Did it help you to understand the parameters of your
Yes,
topic?
it helped much more than the article because it actually showed me how growing organ works. And also, the YouTube videos included interviews that other reporters did with the scientists. So that was very helpful. And over here, moving on to the evidence of primary research. Primary research, I include the evidence to show that it's much harder to acquire than the secondary research where I can just go online and find them. I had to distribute the email to my school and send out emails to invite interviewees for an interview and make appointments. And over here is just my sample survey. It's quite compact, but afterwards I managed to collect the data and make it into a raw table, which then I used to formulate my survey analysis part. And also it included post-research reflection and evaluation, which I added after my folio to go back and evaluate how well I did.
So I wanted to highlight this point that in your survey you just didn't write words, you added also cartoons.
Yeah.
Can you explain to me why you did that? That's
The
unique.
cartoon is just for humor, and also for my distribution of survey, I included a cute pig photo just to get people interested. And also, I talked about what is this all about to just briefly introduce the topic. Because the topic is new and quite complicated, I was afraid that the surveyees might not understand what I'm talking about and therefore give an inaccurate answer.
So for those who don't understand, can you explain to us what is the role of the pig and the chimeric pigs?
Chimeric pig is just basically, since pig, the size of their organ is fairly similar to human, if we put our cells inside pig embryos, they will grow out with our human organs. And afterwards, when they grow to full size, we can harvest their organs and transplant it to human.
When you mean harvest, you mean
Kill. I guess.
kill?
Yeah.
That's it. So that's why you wanted to have some kind of impact on the readers of your
Yeah.
research.
I talked
Lovely.
about it and just talked them, explained that the pig will have to be killed in order to harvest the organs. And I guess this, before doing the survey, the surveyees will have an understanding of what's it all about and develop some kind of understanding towards the topic.
So they would answer being informed.
Yes.
However, your findings there, so what? Do they care if they will eat that chimeric pig?
No, they don't care. In the end, surprisingly, majority of the surveys said they consider the chimeric pig as just purely pig, and after harvesting the organs for human use, they will still eat it, which is weird, I guess. But it helped to contribute towards my outcome.
So that was your opportunity to write a unique outcome?
Yes. Yeah, it was included in the ethical consideration part of my outcome, where I could link my primary research with my secondary online research and cross-reference them. Because my survey included sections where surveyees can write their own elaborated answer, I got a lot of elaborated answer back, and they were sort of small interviews, which I used in my outcome as well, and they were fairly helpful.
So what's this page now?
This page is my interview, primary source, interviewing 2 organ transplant patients. Because in the survey, I distributed the survey with a topic to people who probably didn't have a past organ transplant, and I believe their view on the topic might be a bit biased because they don't know how it feels. So I found 2 organ transplant patients, contacted them, and organized an interview. And in the end, their opinion reflected the data I got back from the survey, where they said they believe the chimeric pig can also be used as food source, and they would accept the organ grown in a pig. So that just further proves that I guess people really don't care as long as the science can save them.
That's a very important ethical component, eh?
Yes.
They can eat, they're happy to eat, the pig they got the heart from.
Yes, and especially when they grow organs in pig, they put their own cells in. So in a sense, they're eating something that has their own cell, which is cannibalism, I guess. And moving on to personal reflection. This is an entire page of reflection, similar to the small parts beforehand, where I just reflect on what worked and what helped me out in writing my outcome.
Okay, so what's this last page?
That's my action plan, where I attempted to follow to finish my research project, but the action plan was very theoretical, or it's not very accurate in the end. I did not follow it on the dot, so therefore I annotated them and added a section on post-research reflection where I talked about which part of the action plan I followed to the point and which part I had to change, and also what change I made from when I began my research project to when I finished, and also what I would like to improve if I redo my research project.
Great. And all this helped you in the writing, the evaluation? Yes. Now, your outcome.
Moving on to my outcome, it has a front page, but this is just starting from an introduction. I did a short introduction, just a couple of lines talking about the topic. This way I don't waste the word count on explaining in the introduction. And I quickly moved on to the main topic of growing human organs in animals. In this section, I talked about the science and how scientists can put human cells in chimeric pig embryos.
So the technology behind this?
Yes, the technology.
Okay.
And I thought it might be quite hard to understand. I also included a diagram, a simple diagram of the process and procedure. And these information were found mostly from secondary sources. Therefore, I included quite a few footnotes in the first page. Moving on, from my secondary research, I realized pig was not the first animal to be chosen to grow human organs in. The first one are apes and chimpanzee, which have a more similar DNA to human. But however, they quickly refused to grow any human organ in chimpanzee, especially in Japan, where the scientists believe they're similar to human, and killing them would result in something unethical. Therefore, I included a small timeline, jotting down the process of this science, where it's up to. So far, it's in progress on testing on non-human primates, such as chimpanzee. Although these organs are not transplanted into human, they're grown into pig and then transplanted into apes to see how well they work.
So instead of transplanting to humans, they transplanted to apes
first.
And if the ape was alive, then could stay alive, then they could try that on humans.
Yes.
Okay.
Right now this is in progress, but afterwards they can either try it on human, or otherwise only try to bring some pig cell into human and see how human will reject the cell, which will be much safer, but it will take a longer time until this science can be used.
And this is...
Moving on to the ethical issue.
What is this page? This page there?
This page, yeah. This page, I talked about the hypoacute rejection, which is pretty common once you try to mix animal DNAs with humans. Hypoacute rejection is basically within 24 hours, the new organ from an animal will be rejected by a human, and immediately the organ would rupture and break down internally. So to better explain it, I included two actual photos of kidneys. One is one that's being rejected and one from the healthy pig. And the one that's being rejected just looks unhealthy and mottled. Moving on to PERV infections. PERV infections, PERV is inactive provirus, which is in the pig cell. They've been in the pig cell for thousands of years or even more than that, and it doesn't affect